Hello, good afternoon, everyone. We will be finishing up lecture 4 today, and hopefully, we will have some time at the end to talk about the lab, which you already have access to, but it's not due until next Wednesday. All right. Last class on Monday, we were talking about the ATP and the ADP. So just to quickly recap, ATP is the usable form of energy in a cell, and it contains 3-phosphate. When you want to use the ATP, you will break off one of the P's, one of the phosphates, and that will convert it into ADP. So ATP is the fully charged version, and the ADP is the drained version. But once you use it up, you can recharge it. You can add the phosphate back on, and the energy that you use to recharge it comes from breaking down the food that you eat. So uh, all day long, ongoing is basically a cycling between the ATP, using it for cellular work, and then it gets converted to ADP, and then you will recharge it back to ATP. So it's just switch between the two forms. It's like charging our phone. Okay, we charge it at night. Maybe you charge it at night, okay, and then you use it throughout the day, and then you drain the battery. You got to charge it again, and then day in and day out, it just switches between fully charged and then using it, and then that's exactly what's going on here with the ATP and the ADP. Now, what I did not tell you on Monday is what exactly do we use the ATP for? What we know we can use it to do um, cellular work, but what kind of work does that include? So we can use ATP in the cell to achieve mechanical work. So mechanical work basically means some kind of movement is achieved. So maybe moving the tails of the of the of the cell, right? Like in a sperm cell, for example, it has a flagellum. To beat that, to create motion, that requires the use of ATP. When we move things across the cell membrane, sometimes we need to use ATP as well, and that's called transport work. And in some cases, when we do chemical reactions, we need to supply ATP to make up for the difference in activation energy in some instances. So the key thing to know here is, regardless of what kind of work we're doing inside the cell, they all use ATP. And when you use the ATP, it gets converted into ADP plus a free floating phosphate. That phosphate, once again, can be added back into the ADP to regenerate ATP, as long as we are eating food and breaking them down to extract the energy from them. Okay. There is a question here. We talk about metabolism being broken down into. Oh, actually, I think we did this question already towards the end of last class. The answer are B and C. It's in the end of last class. You can check the recording if you want. All right, now we're gonna talk about different types of transport, right? Specifically how we move things in and out of the cell. And we have 3 pictures here of red blood cells. We're gonna go through the different cases that would result in these conditions for the red cells. But first, let's start with something simple. We have diffusion. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration. If you think about a glass of water and then you add like 5 teaspoons of salt and dissolves it, that's a concentrated solution. That's a high concentration because it has a lot of salt in it. You take the same amount of water in another glass, but now you only put like a pinch of salt in it, then that's low concentration. Okay. So when two areas are connected and there is a difference in concentration, naturally, material will move from high concentration to low concentration. So here, if you add a few food dye into a flask of water, It's high concentration here, and then it's low concentration here. We use square bracket to represent concentration, okay? So even if you don't touch the flask, you let it sit there, then slowly the dyes are gonna move throughout the flask, and eventually the whole thing is gonna be blue. Okay, so that is called diffusion. Okay? This happens spontaneously. Right? That means it happens on its own. No energy is required, and things will naturally move down its concentration gradient. Okay, so concentration gradient means the difference between, the difference in concentration between two areas. Okay? So in this case, there's a high concentration here and low concentration here. There is a gradient. So the material will go from high to low again on its own. So that is diffusion. Pretty straightforward, I hope. Now, how fast diffusion happens, or we can say the rate of diffusion, is going to be affected by different things. And I'm going to tell you about 4 factors that will affect the rate of diffusion. First, the size of the molecule. If you have something that's small, like this one, this is oxygen, just 2 atoms, that's going to be able to diffuse faster than something bigger, like this glucose molecule. Okay? Things that are smaller tends to be able to spread out quicker than bigger molecules. Second factor that affects the rate of diffusion would be temperature. The higher the temperature, the faster things will spread out, because higher temperature gives more energy to the molecules, and they become more energetic. They're able to spread out quicker. On a hot summer day, sometimes if you just walk on the street and it's like garbage collection day or something, you might smell the garbage, okay, very easily. But on a cold winter day, same street, same garbage collection day, you barely notice the odor, right? That's an illustration of how temperature is going to affect how fast diffusion occur. On a hot summer day, the smell diffuse quickly, and everybody can smell it. Um, Not so much on a cold day. Next, we have the steepness of concentration gradient. So I wrote down for you the definition of concentration gradient a little bit a few slides ago. It's the difference in concentration between two areas. So for example, in this picture on the left, I'm just making these numbers up, okay? Let's say this is 100% concentration because it has a lot of them, right? Okay. And over here, there are nothing, so we'll say 0%. So the difference between them, between the two area, is 100. And these things are going to go through to the right side very quickly. On the other hand, if you have, let's say here is like 80% and this is like 40%, the difference is only 40 as opposed to 100. They will still go from high to low concentration, but it's not going to be happening as quickly. Right? So all these factors I'm describing to you talks about how fast diffusion is going to be happening. Okay. So the greater the difference between two area, the faster they are going to diffuse. And the last factor that affects rate of diffusion is the charge of the particles. Okay? So the key thing here to remember is that like charges repel each other. Like charges repel each other. So they're all positively charged. They don't like to be next to each other. So they will push each other away, right? As far away as possible. So that repulsion between the particles speed up the process of spreading out. Okay? So 4 factors: size, temperature, steepness of concentration gradient, and then the charge. Anybody have any question? Okay, we're gonna take a note here. We actually have quite a bit of note taking today. Beginning with osmosis. So we just learned about diffusion, which is things going from high concentration to low concentration. And osmosis is, you can think of it as like diffusion of water. Diffusion of water. More specifically, you can think of like the more proper way of defining osmosis is the movement of water across a selectively permeable membrane. So whenever a membrane is selectively permeable, it means it's not gonna let everything through. There's some regulation going on. And if we focus on the movement of water across such membrane, then it is called osmosis. So let me draw this out for you. And it's important for you to understand this. Okay? This next concept here is gonna determine whether you understand the rest of today's lesson. Okay, so make sure you ask questions if I'm not making sense to you. Okay, there are no bad questions. If you have a question, I guarantee you someone else in the class has the exact same thing. So be sure to ask for help and clarification. So here we have a U-shaped tube. It's open here at the top so I can add stuff into it. And at the bottom, we're gonna put a membrane. Now this membrane is special. This membrane only allows water. Only allows water to move through. Nothing else goes through except for water. These kind of things exist in real life, okay? People who do dialysis, like if they don't have a functioning kidney and they need to go clean their blood through dialysis, these kind of membranes are used all the time. Okay? So let's say we have a membrane at the bottom that only allows water to pass through and nothing else. So we're gonna get some water, people, and we're gonna fill this thing up. Okay, let's fill it up. There we go. So, you add some water here, you add some water here, it doesn't matter. We just go on both sides, and then it will fill up the container here. Okay, same water level on both sides. You guys good? You know what I'm talking about here? No confusion with the drawings or anything like that? Okay, so next I'm gonna get some salt and then I'm gonna add it to the left side. Okay, you should probably be doing this in color as well. Right? Otherwise, it's like not gonna make much sense to you. Right, so I'm gonna add some salt here, and then I will also add some here, but not as much. So these are solute. And I mean, they can be anything, but we'll just say it's salt, okay? Salt. So I want to know which sign has higher concentration. Let me just do a poll here. Which sign has higher concentration? Is it A, this sign, or B, this sign? Both sides has same amount of water, but different amount of salt. Which sign has higher concentration? It's not a trick question. Okay. Yeah. Okay. Most people got it right. The left side, because it has more salt in the same amount of volume, this has higher concentration. High concentration. And here is low concentration. Now pay attention to this next part here. This is where it gets confusing for people. We see high concentration on the left and low concentration on the right. The red dots, the solute, they would want to go to the right side because things naturally goes from high concentration to low concentration, right? So the red dots want to move to the right. Can they do it though? Can the red dots move to the right? What do you guys think?
No.
No. Okay. Because we have a barrier at the bottom, right? This barrier blocks it. The red dots are too big to fit through. Okay. So even though they want to go to the right side, they can't. It's blocked. Okay? So the only thing that can move in this situation is water. You got to understand, in nature, things tend to want to balance themselves out. Okay. Like if the concentration here on the two sides are not the same, then there will be some kind of movement to try to balance it out. And the only thing that could help with balancing it would be the water. Okay? So which way will water move? Which way does water move? Remember, the red dots cannot move. So what do you think? Is it going to move to the left? Is it going to move to the right? Let's quickly do a poll here. I'm going to tell you it's going to move, but you think about which way it is going to move. Okay, thank you, Sophia and Mary. And everyone else, if you see the poll, please choose something. If you don't see the poll, you can type your answers in the chat box, just so that it's more for me than it is for you, okay? Because I can't see your faces. I can't see that, usually I can read the room and see whether people understand this stuff or not. By showing me what you think the answers are, it'll help me see whether I need to explain this in more detail. Okay. Last call. Anyone else? Okay, so we most people say left. We have a few people who says to the right. All right, so here is the thing. I want you to go through this mental visualization with me. Okay, imagine, imagine taking a spoon and you scoop up a nice spoonful of salt, just like salt from the kitchen. Okay, big spoon. You're gonna put it in your mouth, and then you're gonna keep it there. Okay, close your mouth. Don't swallow, just keep the salt in the mouth. Okay, imagine doing this. And you hold it there for like half a minute. Okay, what's gonna happen after 30 seconds, after half a minute? How is it gonna be looking around your mouth, for example? How does it feel in your mouth? Can you visualize this? I mean, you can always try it later, but okay, it's gonna be yeah, dissolved liquid. Okay, anyone else? Spoonful of salt. Hold it in your mouth. Don't swallow. What is it gonna be like? Well, if you haven't tried this before, maybe try it out to help you understand what's going on. But I can tell you, if you do that, then you're gonna start drooling, actually. Okay, you will feel a lot of saliva is being sucked out of your mouth, and then you're gonna be like drooling all over the place. Um, and that illustrates how water tends to be attracted to an area of high concentration. Okay? The salt essentially draws the water towards itself. And that's what's happening here. Okay? Because you have more salt on the left side than the right side, then the water is going to be attracted to the left side. And as water moves to the left, as water moves to the left side, the left gets diluted. Okay? Because you're adding more water. The water is going to move this way. The water level is going to go up. The number of red dots don't change. You are just adding more volume, and that's going to dilute it. Okay? If you're having trouble understanding this concept of dilution, think of like some kind of orange juice or something. If it's too sweet, you can add more water, and it becomes less sweet, right? It becomes more diluted. While the right side while the right side gets more concentrated. Okay, because there are less things here, less solute, then the water is going to move to the left, and then the water level is going to go down. And it's like reducing a sauce when you cook. When you evaporate the liquid, it gets thicker and thicker, and then it gets more concentrated, right? This will continue. This continues until the concentration on both sides are equal. There is a question, does it work to make the right side more neutral? Can you elaborate your question? I don't quite get it. Maybe explain a little bit more. What do you mean by make the right side more neutral? Oh, I answered it already. Oh, okay. Yeah. All right. So as the liquid goes to the left side, the water level on the left is going to go up, the water level on the right is going to drop, and eventually the two sides will have the same concentration. So this is osmosis. Okay, the focus is the movement of water. Please let me know if you have any questions, because we're going to be taking another note that's going to be like 25 minutes long. And you got to understand this concept for that note to make sense to you. So I'm just going to open up the floor right now, I guess. That's what they say. And see if anybody want to say anything, ask anything. It's all crystal clear, I hope. All right. So this is another picture, right? That shows what happens, okay? Of course, in this picture is opposite to what I drew for you. I did it on purpose, okay? So you can see the two different scenarios. Here, the water is going to be drawn to the right, and eventually the water level will rise on the right, and it will drop until both sides are equal. So that's osmosis, all right? That's how people preserve food before refrigeration was invented. Right? To prevent things from going bad, they salt the food. By salting the food, you're basically using the salt to remove the moisture. Okay, you cure the meat, you cure the fish, and without the moisture, the bacteria can grow and they last longer. Okay. So excellent way of preserving food, but all that extra sodium, it's not too good for your health. But now you know why it works. All right. Next, we're gonna take a note on this thing called tonicity. Tonicity. And it's a long note. If you have a you will need a brand new piece of paper, okay? New piece of paper. You're gonna separate it into three areas, okay? And then three columns. Like you don't have to draw the lines, okay? Like, but visually you think to divide it into 9 boxes. So we're gonna draw picture, 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 and then we're gonna write words in the middle. Okay? So that's how you plan it out. Already. Tonicity. Tonicity. So basically, tonicity is the ability of a solution to change the volume of a cell by altering its water content. Okay. That's what the word means. A solution, as you may recall, is basically what you get when you dissolve a solute in a solvent, right? Adding salt to water, you get a salt solution. Okay? In the body, of course, is more complicated. It's not just salt that we're dissolving. There are like tons of other stuff that are dissolved in a lot of our bodily fluid. Um, but to keep it simple, just think of it as salt for the time being. And when we learn about blood next semester, I will tell you, you know, all the stuff that you will find inside the blood. Okay? For now, just focus on the salt. All right, so we have three situations here. First situation, first case, it is called isotonic. Isotonic solution. Okay, we're gonna draw a before, and we are going to draw after. Before and after. So here we have a beaker. Beaker. A beaker is just like a glass container, okay? And we're gonna put a cell in it, an animal cell. Obviously, the cell is not that big, okay? Like if you actually put a cell in a beaker of water, you won't be able to see the cell. Okay. So that is an animal cell. Okay, thanks for letting me know. Okay, feel like I got your message. And here we have another beaker, and we will put a plant cell in here. Plant cell. Now, plant cell in general, because of the cell wall, is gonna have more of like a rectangular shape compared to the animal cell. By the way, for this nook to look better and easier to understand when you come back and study it, please use some colors, okay? You need like 4 colors maybe. Okay, so in an isotonic solution, it means the concentration of the solute are the same on the inside and the outside of the cell. Okay, they're the same. Same concentration on the inside and outside. I'll write down for you. Concentration, which is what the square brackets are. Concentration of the solution, which is like the surroundings of the cell, is going to equal to the concentration of the cytoplasm. Cytoplasm is the inside of the cell, right? So what's the same? Right? Same amount of salt on the outside and inside. So based on what you just learned with this YouTube thing, U-shaped tube, um, which way do you think water is going to move? It's going to come into the cell, go out of the cell, or what do you think is going to happen? Okay, so Alpha says no movement, and that would be correct because it's the same on the inside and outside. So there is no preference as to where the water will go, right? The more accurate way of describing it would be it's not that the water is not moving. However, the amount of water that's moving in is going to equal to the amount of water that's leaving the cell. So overall, they cancel each other and there are no net movement, but the water is still coming in and leaving. So let me write that down for you. Rate of water leaving the cell equals to rate of water entering the cell. No net water movement. Okay. Like if someone spends as much money as they earn, there's no net income, right? Water coming in, same amount leaves, no net water movement. No change in volume. Now, to help you understand, we can use an analogy, okay? We have a bucket that has a hole at the bottom, and we're gonna have a tap here, okay? And we will turn on the tap. Right? There's some water in there already, okay? We will turn on the tap, but then water is leaking at from the bottom. But if the water coming in at the same rate as it's leaking, then your water level will not change. Water is still moving, it's coming in and leaving, but overall, the water level is stable. And that's what's happening here. So afterwards, what we see? What would happen? Nothing happens. Animal cell is going to stay the same. I'm not going to draw the dot because I don't want to confuse you. Plant cells going to be the same. Okay. So for animal cells, they are happy. Okay. Our body cells prefer isotonic solution. Okay, keeps them happy. For plant cells, they're not happy, but they're not sad. Okay, they're like, it's all right. Okay, I'll live. It could be better, but it's all right. It's not the worst. They're like meh about it. Okay. So that's like, if you forget to water your plants for 1 or 2 days. Okay. Obviously, some plants don't need as much water, but generally speaking. So that's it. That's the first case scenario, isotonic solution. Does everybody understand? That's the easiest one. I have two more for you. Okay, there's a private message question. So the cells has no mass to disrupt the water movement. I don't 100% understand what that means, but the cell in this case is doing it's more passive, okay? The water is coming in and it's leaving, and they kind of don't have a way to regulate it, okay? Um, so they're doing more like a passive role here. It's just something that occurs. I don't know if that answers your question. If not, feel free to rephrase it, please. I'll get back to you. Next, we have another scenario, which is hypertonic. Hypertonic. Okay, solution. Hyper means too much. Okay, too much. That's what hyper means. So let's do the before and the after. And we are going to draw the beaker again. This is just water. And after it, we'll see what happens afterwards. But then we're gonna do another one here, another beaker. Okay, we're gonna put our lonely animal cell in there and the plant cell. And this time it is a hypertonic solution. So hyper means a lot. So there are a lot of solute on the outside. Okay, just a little bit on the inside, but a lot on the outside. So help me out here, guys. Which way do you think the water will move? Into or out of the cell? Okay, very good. So let's write down the stuff first. In this case, the concentration of the solution is greater than the concentration of the cytoplasm. Okay, greater than, okay? I think you learned this in math, but just in case, that's greater than. And so you will have water coming in, but more will leave the cell because the water is attracted to the saltier side. So it's not that the water's not coming in, but it's just not as much coming in than leaving. So in this case, uh, what did I say before? Rate of water leaving the cell. Okay, so rate of water leaving the cell is greater than the rate of water entering the cell. So overall, you have net loss of water. Okay, let's draw our leaky bucket again and see if it makes any sense at all. There is our bucket. We have some water in it. And this time, we are keeping the tap the same, but we have a really big hole in our bucket, and it's like leaking all over the place. Okay, so yes, water is coming in, but you are really leaking at the bottom. So what's going to happen to the water level? Well, common sense should tell you that the water level is going to drop, right? So if a cell is experiencing a net loss of water, what's going to happen to its volume? Does it get bigger or smaller?
Ball.
Mm-hmm. So cell volume decreases. It shrinks, basically. Okay, so for an animal cell, afterwards, it is going to shrivel up. Right? It's like a grape, except grape is not animal, obviously. But like if you remove all the water from a grape, you end up getting a raisin, right? It's like a shrivel up. So we have a special name for this. It's called plasmolysis. Plasmolysis shrinks. Same thing for the plant. It's gonna shrink, but the cell wall is not gonna shrink, okay? But the cell membrane is gonna kind of pull away from the cell wall, and your plant cell is like, all shrivel up on the inside like that. Okay, so that's the cell wall. It's still pretty much the same thing, but the cell is shrivel up. It's also called plasmolysis.
Is it only the vacuole that will be?
Yeah, exactly, right? So the central vacuole is now drained, and then, it's kind of deflated the cell. Okay? But the cell wall stays where it is. Very good. So this is for us, okay? We're like, okay, it's not the best, but we'll be fine as long as we drink water soon. All right. So if you're dehydrated, if you haven't been drinking all morning, all day, I mean, today's not bad. Today's like, pretty chill outside. But like on a hot summer day, if you're sweating a lot, you're not rehydrating yourself, then your cell will be shrinking. They will be plasmolyzed. We're not happy about it, but we will live. Plant cells not good. Okay. This is like forgetting to water your plant for like, a week or more. The leaves is really like shriveled up. They might even dry up and start falling. Plants don't like this. You have a part in the brain called the hypothalamus. We'll learn this next semester in the brain unit. The hypothalamus will shrink when you are dehydrated. The shrinking of the cells in the hypothalamus is going to trigger the thirsty sensation. Okay? So when you drink water, you dilute the surrounding of the cells, and then you restore the isotonic state. Okay, so that's that. Last scenario is going to be hypotonic. I want you to put a big O here, okay? Hypotonic solution. Hypo is too little, right? Or under. Like if someone has like hypothermia, their body temperature is too low, right? So in a hypotonic solution, you probably can guess what the drawing is like. What am I doing? Okay, this is after. Sorry. Um, there you go. We're gonna have our beaker, and we will pop in our animal cell, and we will do the same thing for the plant cell. Plant cell like that. So this time, hypotonic, it's below, as in the solution has less concentration compared to the inside. And again, I really hope you're doing this in color. And if you're not, please redraw it later with color. It will make so much more sense, okay? Otherwise, it's like really confusing, I would think. So in this case, you would have more water coming in. Remember, water is attracted to the more concentrated location, more concentrated area, which is the inside of the cell. So more water comes in than leaving. So the solution concentration is less than the concentration of the cell, of the cytoplasm. Cytoplasm is the inside of the cell. And therefore, the rate of water leaving the cell is less than the rate of water entering the cell. Okay. And then what's going to happen is you have net gain of water. Cell volume increases. let's draw our analogy here. Gonna have our bucket again. It's leaking normally at the bottom, but this time we're gonna turn on the tap all the way to maximum. Lots of water coming in, some water leaving. So the water level will rise. Eventually it will overflow. Okay? That's not difficult to visualize. And so for our animal cell, afterwards, it's gonna basically, like if you let this happen continuously, it's gonna swell up, swell up, swell up, and eventually it's gonna pop. So the animal cell will pop. Right? It's full of holes now. We call this lysis. It will burst. What about the plant cell? Do you think the plant cell will burst? Okay. I see a few yeses, some nos. Well, the plant cells have something that we don't have, right? The plant cells have a cell wall. So the cell wall will actually protect the plant cell. Okay. So it will swell up with water. It will get really, really plump. Okay. And then the central vacuum inside is going to be very, very filled up. And it's going to create some pressure that pushes on the cell wall. So the plant cell will not burst. It will just get really, really, really like puff. Okay, like you think of a Ziploc bag that's really full of water. Okay, you touch it, it's like, ooh, there's pressure in it, right? So this is called turgid. Okay, and this turgid pressure will help the plant stand up against gravity. The leaves are gonna be like all opened up. Okay, and the plants are very happy. Okay, so the best state for a plant to be in is a hypotonic environment. For animal cells, nope, we don't like it at all. Okay, it's dead, basically. Okay, that's that. Now I want you to understand these terms are relative terms. Okay, let me give you an example. If I say I am taller than you, I am taller than you. Another way to say the exact same thing would be you are shorter than me, right? You understand that? If I'm taller than you, that's the same as saying you are shorter than me. Like, how does this relate to what we're talking about? Over here, we are using the hypotonic and the hypertonic to describe the solution. So when we say the solution is hypotonic to the cell, another way to say it would be the cell is hypertonic to the solution. Okay, one more time. The solution is hypotonic to the cell, but the cell would be hypertonic to the solution, right? Just like taller and shorter. So generally, when we talk about a question, when we do a question, we're talking about the solution. Okay, but just keep in the back of your mind. Sometimes we can describe the situation from another perspective, and the terms will be switched. Okay. So the reason I put a big O here for hypo is for you to remember when you see hypo for the solution, the cell swells up. It gets really, really, really big. If it's a plant cell, it's gonna pop. If it's an animal cell, it's gonna pop. If it's a plant cell, it's gonna be okay. That's pretty much it. Let's take a look at this picture here. So we have the isotonic environment for the cell. These are red blood cell, by the way, red blood cells. They help carry oxygen and carbon dioxide in the water and in the body, I mean. So if it's isotonic, they are happy. This is the normal shape. If it's hypotonic, they swell up. Right? They might not burst. Like this is, if you just have like a little bit too much water, you might be okay. But if you have like really a lot of water, then it's gonna swell up and burst, and it's not good. Hypertonic would be like shriveled up. Okay, and as soon as you rehydrate, it's going to be okay again. Uh, some of you might know there is a video console called the Wii, which is like Nintendo. It's like more than 10 years ago, right? But when it first came out, it was like really popular. Okay, a lot of people want to buy it. And so in the States, there was a radio station that hosted a contest, and it was called like Hold Your Wii for a Wii. Okay, that's the name of the contest, and as the name suggests, contestants had to drink water and not go to the bathroom. And if you're able to be the last one standing, then you win the video game console. Okay, so this young lady, she won the contest. Okay, she was able to drink a lot of water, and she held it, and then she won the video console. But then she was found dead in her apartment a couple hours later. So what happened? Well, based on what I taught you, it's perhaps not surprising. With a drastic increase in water content in the body in a short period of time, you basically dilute your blood to a point where it becomes hypotonic. And when it does becomes hypotonic, then your red cell starts to pop, right? And as I told you, red cells are important for delivering oxygen. If a substantial amount of red cells die, you won't be able to transport oxygen anymore. And that's how you could die. It's called water toxicity. Okay, you can look it up if you want to learn more about it. Okay, moving on. Okay, I was gonna tell you about IV solutions, but honestly, we are kind of running tight on time, so I'm gonna skip through these parts. I will not test you on it. Okay, so no IV solutions, but it's good information. Because if you're gonna become like a practical nurse or something, or like a registered nurse, you will have to give people IV solutions. And to know what kind of IV solutions to give, it's actually related to what we're talking about. You can give them normal saline. You can give them hypotonic or hypertonic saline, and it all depends on what the medical situation of your patient is in. Okay, so you can read through that on your own. I don't have time to go through it, so we're just gonna move on. All right. The last note we are going to take will be on the type of transport. I'll basically reconstruct this whole thing for you. I mean, I could read through the PowerPoint with you, but usually, again, like if I do that, things just tend to go in one ear out the other. I think it's much better if we draw it out together, and then you can read the PowerPoint slides later to fill in the extra information and whatnot, right? Okay, so please take out another piece of paper if you can. This time, I would suggest you do it sideways. We will start here, and then we're gonna separate it into different categories, and then it's just gonna go down all the way to here. Okay, so not super big first, because we have many layers we need to go into when we draw the note. Okay, definitely try to do it sideways, please. So this is transport. Transport across the cell membrane. Okay, that's what we're talking about. Moving things across the cell membrane. So there are two kinds of transport here. Let's start with passive transport. Passive transport. Okay, I'm gonna skip the break today. I'm very sorry. I just want to make sure that we have enough time to go through the lecture and then tell you about the lab. Okay, but no class next Monday. Remember, okay, college is not open next Monday. All right, so here's the passive transport. What does it mean by passive transport? In passive transport, things go from high concentration to low concentration. Okay? Diffusion is an example of passive transport. It goes from high to low. It happens spontaneously, so no ATP needed. Okay. Just occurs on its own. You don't have to supply energy for it. It's like a ball rolling down the hill. Just happens on its own. That's right, osmosis too. On the other hand, we have active transport. Active transport. This is going from low concentration to high concentration. It's like rolling a boulder up the hill. That's going to require energy. Okay. Requires ATP. Okay. So the first thing I told you today is that we can use ATP for various things in the body. Mechanical work, as in moving body parts. Transport work, as in active transport. Okay, so if we want to move things against their concentration gradient, we need to do active transport. So let's take a look at different examples of passive and active transport, starting with passive transport here. Okay, so we already know that, we already know an example of passive transport is diffusion, or sometimes we call it like simple diffusion. Okay, because there is another kind of diffusion that I will tell you shortly. This is simple diffusion. Simple diffusion is best for small molecule. Okay, small molecules that can squeeze through the cell membrane on their own. So I'll put some examples in red, such as water. Okay. I know water is also involved in osmosis, but osmosis is a type of diffusion as well, right? Also, if it's hydrophobic molecules, it's going to be able to go through quite easily because if you remember the structure of the cell membrane, there is a thick layer of hydrophobic tails in between, right? So if a molecule is hydrophobic, it will have no problem crossing that thick layer of hydrophobic tails. So things like steroids, okay, they will be able to go through no problem. Okay, like steroid hormone. Right. Last class, I told you about the different functions of membrane proteins, right? One function, the second function that I told you was the cell signaling. Okay, so if some molecules are too big, it cannot go in to create the signal, then it will have to bind to those receptors. But if it's a steroid hormone, it's able to cross the membrane and it's going to go directly to the nucleus and change how the cell behaves. Also good for nonpolar molecules. Nonpolar molecules such as carbon dioxide, oxygen, they will be able to go through the cell membrane no problem. In fact, that's what happens every time you breathe. Okay, air goes into your lungs, the blood picks up the oxygen. That happens through simple diffusion. Okay, so I'm going to add osmosis here as well, which is like diffusion of water, right? These are like related. Okay, so I'm just going to put like water again. Other things are not called osmosis. If it's other things that moves, then it's like simple diffusion. All right. I'm going to add another one here. I'm going to drag it down all the way here. Try to follow what the shapes are so that you don't run out of space. This is another kind of diffusion called facilitated. Facilitated diffusion. Let me give you the definition first. Facilitated diffusion would be movement of molecules down its concentration gradient with the help of a membrane protein. Okay, I'm just gonna pause and let you catch up. You're gonna give me some thumbs up if you guys are ready. Sorry, just replying email here. Okay, hopefully you all caught up. So it is still passive transport we're talking about. This still does not need energy, and it's still going down its concentration gradient from high to low. But it's too big. Okay, the molecule might be too big, or it might be charged. So it needs a bit of help crossing that thick layer of hydrophobic tails. And there are two kinds of facilitated diffusion here. Right? So this first example here is called a channel protein. Channel protein. And channel proteins, they are best for moving ions. Right? Remember, ions are charged particles, right? So example, if you want to move some calcium into the cell or out of the cell, then you're going to use use a channel protein. And this is what they look like, right? So I'm going to draw the phospholipid here. Okay, not going to go crazy, just going to have 6 on each side. And a channel protein is basically an integral protein. Give me 1 second. It's an integral protein that literally has a tunnel in the middle, has a channel in the middle. Okay, so if you have some calcium here, calcium, 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 you have some on the inside as well. These calcium cannot cross the cell membrane on their own because the hydrophobic tails will block polar stuff, right? So they would go through this tunnel and enter the cell. The channel protein basically provides a tunnel to shield the ions from the hydrophobic environment. Okay, so they can just flow through. Okay, you gotta understand it's not necessarily coming into the cell, right? If there are more calcium on the inside, they might end up leaving the cell as well. So that's channel protein. And then we have carrier protein. Both of these are examples of facilitated diffusion. To facilitate someone is to help someone, right? So facilitate diffusion basically means the membrane proteins are helping these molecules to cross the membrane because they cannot do it by themselves. Okay, so I'm going to draw another one here. Okay, I'm just going to draw an extra one here as well. Okay, so it looks something like this. It's going to be it's like Pac-Man. You know Pac-Man, like that video game where the yellow guy eats all the dots, right? So it's like Pac-Man here. And let's say you have a really big molecule, okay? I should write it down for you. Best for moving big molecules. Big molecules that are not hydrophobic specifically. So like glucose. All right, so let's say that we have the glucose right here. It is going to bind to the carrier protein. And then the carrier protein will change shape such that it is now facing the inside. And then it will spit out the molecule. Okay. Another way to think about this is like those, those revolving doors sometimes they have at the mall, right? Like you hop in on one side, you push yourself in, and then you exit, right? So it's like a revolving door. Right? The difference between the channel protein and the carrier protein is that the channel protein doesn't change shape. Right? It's just a tunnel, and you walk through the tunnel. But the carrier protein, it keeps on flipping back and forth, right? And every time it flips, the opening is different. Right? So it grabs on to the molecule, it flips, and then it opens up, let it out, and then it flips back, grab another one, and so on and so forth. Okay, so both of these scenarios does not require energy, and they are both example of um, facilitated diffusion. Okay, that's all for now for passive transport. Anybody have any questions before I move on to active transport here? Okay, active transport. Active transport requires ATP. We are moving things uphill, so to speak. We are moving things against their concentration gradient. Okay? So there are different kinds of active transport depending on what you are moving. So one example is the protein pump. So this is, once again, a membrane protein, and they use ATP to move things against their concentration gradient. Uh, so, for example, we have something called the sodium-potassium pump. Sodium, this is an example, okay? Sodium-potassium pump. Many cells have the sodium-potassium pump, especially important in the neurons, and this is what it does. Okay, let me try to draw it out for you to help you understand. These are phospholipids. And we have the sodium-potassium pump here. All right, so it will have slots for potassium. So there are 3 potassiums here. Okay. And then it will have slots for sodium. And this is what happens. It will eject the sodium. Actually, sorry, I wrote it the other way around. Give me one second. Let me just move this out of the way. Okay, so the reds are actually the potassium. It will bring in 2 potassium ions, and in exchange, it will take out 3 sodium. That's what the blue balls are. Okay, those are the sodium. So over time, you will have a lot of sodium on the outside and then a little bit on the inside. On the other hand, you will have quite a bit of potassium on the inside. Okay, the squares are the potassium, okay? And then you won't have as much on the outside. Okay, this is outside of the cell. This is inside. So why does this require energy? Well, if you think about it, if you keep on bringing potassium in, then you will have high potassium on the inside and low potassium on the outside. Similarly, if you keep on pumping sodium out, you will have high sodium on the outside and low sodium on the inside. So this keeps on happening, and you are pumping the ions against their concentration gradient. You're taking it from an area that's low concentration to an area that's high concentration. Same thing for the sodium. That's why you need to pump them over. Okay, it's like pumping water up a tower for the town. You know those big wide water towers, right? Water gets to get pumped up. Right? When it comes down, it doesn't need water, it doesn't need energy. But when you open the tap, the water comes out. There's no electricity required. That's why even when there is a blackout, the tap still works. Okay? But pumping them up, that requires energy. And here we have a protein pump. Okay? I'll give you another example of a protein pump. Is called the proton pump. Proton pump. Okay? Proton pump you will find in stomach. Stomach. Okay? This is a stomach. Okay? You might or might not know that there are acid in the stomach. And these proton pumps basically keep on pumping H plus into the acid. Okay? So people who have like heartburns chronically, they might take something called PPI, which is a proton pump inhibitor. Okay, so it blocks some of these pumps, so it reduces the acidity of the stomach acid. So these are examples of protein pumps. They are active transport and they need ATP. We are almost done, guys, with the note. So over here, we have another category, which is exocytosis. Okay, and then next to it, we will put another one called endocytosis. So, actually, I am so sorry. I forgot to put another name here. Oops. Sorry. I can hear you screaming at the screen. This is bulk transport. Bulk transport. And then we have 2 different types, okay? Exocytosis and endocytosis. So bulk transport just means moving large quantity of materials across the cell membrane. It depends on which direction you're moving. If you're moving things out, then it's called exocytosis. If you're moving things in, then it's called endo. exocytosis. So exocytosis is moving materials out of the cell. And endocytosis would be moving materials into the cell. Remember, all these things are nicely written out with professional drawings in the PowerPoint slides, okay? Please go through them again on your own. I'm writing it out because I think it will help you learn it a little bit better. You might disagree, but that's fine. So exocytosis look like this. Okay. Now maybe watch first, and then you can see, how much to draw. Okay, just give me a second. All right. So an example of exocytosis is actually what we learned previously about the, endomembrane system and the Golgi body and all that stuff. So let's say we have a vesicle from Golgi body. In this vesicle would be a lot of proteins. Okay, different proteins. So there are different places this can go, right? And one of the possible paths would be for it to go to the cell membrane. Okay, this is like a series of events, okay? So this would go and it would touch the cell membrane. And then it will actually fuse with the cell membrane. It will become part of the cell membrane. And as it does, the contents are released out of the cell. Okay, so all these things get released. And you visualize that. Okay, it's like, the bubble slowly go and go and go, and then it touches the cell membrane, and then it fuses with the cell membrane. It becomes part of it. And in the process, the materials are released. So we're not just transporting small amount of stuff. It's like a lot of proteins in there, and it all get released in bulk. That's called exocytosis. Okay? On the other hand, we can do endocytosis, which is to bring things in. So it's the exact same concept here. Okay, maybe I'll redraw this for you quickly. This happens like in this direction. But now for endocytosis, it's going to be bringing things in. All right. So it looks like this. All right, so let's say you have some, green stuff you want to bring in. So they're gonna go and cause like an indentation of the cell membrane, and then the cell membrane is gonna close off, and then you pinch it off, and now you have a vesicle. Okay. Now this vesicle, if you remember the stuff from lecture 3, can you please tell me where will that vesicle go? If you're bringing things into the cell and you want to break it down, where do we need to take it?
Lysosome.
The lysosome, that's right. It's going to fuse with lysosome and the enzyme is going to break it down, okay? So that's endocytosis. Now, I apologize, but there are 3 kinds of endocytosis, and that will be it, and then we're done, okay? Again, I am just a messenger, okay? I didn't make these things up, and doesn't matter where you do the pre-health program, you will be learning these things because we are ministry mandated to teach certain things in the pre-health program. So there we go. We have 3 kinds. Okay? First one is called phagocytosis. Phagocytosis means cell eating. Okay? So when a cell wants to eat, wants to bring in like some solid stuff, that's called phagocytosis. An example of that would be a white blood cell and it sees a bacterium. Okay. It checks its ID tags on the surface and it knows it does not belong to the body. So what does it do? It eats it up. Okay. It actually does this. Okay. I am not making this up. Okay. So it's gonna go surround it. Oops. It's gonna go surround it and then it's gonna swallow it. And now is on the inside. Okay. This is called phagocytosis, cell eating. Okay, just a little bit of extra things for you to think about. Well, not think about, but like it's an interesting thing to know. Okay, we'll learn more about this next semester in the immune system. But did you know some white cells, after they eat the bacterium, they would digest it and they would put some of the bacteria stuff on their surface. It's like a trophy for them. Okay, this is for real. Okay? And they would go around and show it to other white cells. Like, hey, guess what? I just killed. I killed a bacterium, and it looks like this. You should go find these guys and clear them out of the body as well. That's how the white cells communicate with each other. Okay, so by putting bits and pieces of the bacteria on the surface, it's showing other white cells what the invader looks like, and then they would go around and kill them. Okay, more on that later in the program. Okay, so that's phagocytosis. Well, if there is cell eating, you probably guess there must be cell drinking, right? Cell drinking. And it's called pinocytosis. Okay, phagocytosis for eating, pinocytosis for drinking. Right? The difference between cell eating and cell drinking is in pinocytosis, you are bringing in mostly liquid rather than like particles. Okay. So an example of that would be like human egg cells. Human egg cells drink from the surrounding. Okay, from its surrounding. Human egg cell drinks from its surrounding for nutrient. Okay, so it's bringing in mostly liquid instead of like a big chunk of bacteria. Okay. And finally, the last kind, it is called receptor mediated endocytosis. Okay, receptor mediated endocytosis. I mean, I don't have like a special name for it. I'll just draw it out for you. It will be easier to understand. So in receptor mediated endocytosis, first of all, we're bringing things in, but we are a little bit picky about what to bring in. Okay. So on the surface of the cell, there are these special receptors. Okay. It's similar to like I mean it's they are membrane proteins, but they're a little bit different from the receptors for relaying signal. These receptors have a specific shape. So only molecules that match the particular shape will be able to bind to the receptors. Okay, like these other ones here, they don't fit. They don't fit. So we will only be bringing in the blue triangles and not the green circles. Right? So there is some selectivity to it. Okay, so only brings in molecules that fit the receptor. Okay, so that's what it means by receptor-mediated endocytosis. Right? You have to fit You have to meet the credentials before I let you into the building, basically. So there you have it. All the different kinds of transport. Um, and once again, all the nice details are in the PowerPoints. Um, and that is it for lecture 4. Okay, I know there are a bunch of concept check questions. Um, I encourage you to try them. Like last time, I'll post the answer for you to check probably later on. Any questions before I talk about the lab? Our study guide answers released today for lecture 4. Yeah, sure, I'll do that. Okay, I will do that right now before I forget, because believe it or not, I always forget. There we go. It's up now. So what's going on next week? Let's take a closer look. Next Monday, like I said, no class because of the National Day for Truth and Reconciliation. And on Wednesday, next Wednesday, you will be handing in the Beetroot Lab. I think you have until the end of the day to hand it in, and I'm going to explain the lab to you right now. And then October 7th, you will have quiz for lecture 3 and 4. Okay. And then yeah, we will have another day where the college is closed on the Monday, so we lose another class. But that's okay. I'll post a review for you for the test, and then that will be it. Okay. No holidays for the second half, so it won't be as rushed in the second half. All right, let's talk about the lab. So let me switch to student view. By the way, I made a mistake last class with one of the questions. I think I was just like rushing through it and didn't read it properly. The answer should be D and not E. All right. Oh, by the way, like the quiz marks are up right now. If you haven't checked your grades yet, please go ahead and do that. You guys are doing quite well. Class average is 76. It's actually the highest of all my classes this semester. So congratulations. I can't return the quiz to you guys though. Okay. For online assessments, we don't return the quizzes because we need to keep the questions kind of secret. We don't want it to be out on the internet and whatnot. So thank you for your understanding. What you want to do is you want to access the lab, and I will post the link for you probably later today or tomorrow. But the lab is actually done as a quiz. Okay. It's not like not like an actual quiz where you will have to lock down the browser and everything. It's just that if I do it in a quiz format, it's easier for me to mark. That's all. Okay. So if you go to the quizzes, you will see that there is the beetroot experiment. Okay, so the beetroot experiment is available to you right now, and it's going to be available until October 2nd, which is the due date. You can hand it in anytime between now and then. Now, having said that, the experiment is not something you can start today, work on it a little bit, and then, work on it again tomorrow and then the day after tomorrow. There is a time limit. Once you start the experiment, once you start the quiz, the lab, you will have 3 hours to complete it. Okay. And just like a real quiz, you can't quit and stop the timer. Okay. So make sure you have at least, 3 hours of me time, lonely time, where you will not be interrupted when you do the quiz, when you do the experiment. Okay. Does that make sense? Okay. So you click on it, and then there are some description here. Okay, I'll explain some of these for you. Okay. So throughout the experiment, the virtual experiment, there will be some video content along the way. Okay. So you need to watch these videos before moving on to the next session. Okay. These videos is basically me doing the lab, and you have to watch it. Okay? What I don't want you to do is like this. This is what a lot of people do, and it's wrong. Okay. They don't watch the video. They just skip directly to the answer to the questions, and then they just Google the answer the questions, and then, copy the answers from the internet. It's not good because the questions are related to the videos. The people who are posting the answers online, they didn't watch my video, so the answers are going to be incorrect. Okay. And I would know if you are copying from the internet because people would start to talking about like, different alcohol solution when in fact we didn't use any alcohol in the lab. Okay. So please, please, please just watch the videos. They are really short. They're like 2, 3 minutes tops, and then you can answer the question. Okay, there is a question. Would this be an open textbook for us? Yeah, yeah, yeah. You can use the textbook and whatnot, use the notes and everything, but all of the answers are in the videos. If you watch the video closely, you will be able to answer them. There are also answers that might be in the lecture notes for some of the thinking questions. Uh, in other words, there is no need for you to search up anything online. Okay, everything is what we've talked about in class, and the answers are all there. If you're stuck, there are help videos. Okay, these are extra help videos. If you forgot, about certain concepts, I made a video to explain those concepts to you. If you watch those, you probably will be able to get the answer as well. Okay, so like I said, the vast majority of answers to the questions can be found within the video, class notes, or previous lectures. It is expected that you will rely on these resources for your answer. Do not search the internet for answer because that will like defeat the whole purpose. And more importantly, it's very likely you will get the wrong answer from the internet source, and then you won't get the good grades, right? Believe me, it's very easy to tell when someone is taking it from the internet or not, because it sounds like way out of like what I expect to see. Okay. And, I've been doing this for over 15 years, so I've seen a lot of internet answers. So I would know. Okay. Now I will start the quiz to show you. Uh, so here, for example, it says background information, and there's a short clip. You just watch it. It's okay, it's not that short. I guess it's 8 minutes long, but it gives you all the information about the Beetroot Lab. It tells you why you are doing the experiment. So after you watch it, I ask you what is the purpose of the experiment. Okay, then you would say, you would type the answer in. The purpose of the experiment is to investigate blah 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 blah. Okay, it's only 1 point, so don't write me a novel. Okay, just like keep it short and to the point. Okay? I always tell students to remember 3 C's when you are answering my questions. Be concise. Okay, don't ramble on. Be correct and be clear with your answers. Okay. So that's it. And then you would move on to the next question. What is the name of the pigment that creates the distinct vibrant red color in the beetroot? Again, don't look it up on the internet. Okay? Watch the video. It tells you specifically the name of it. Okay? Now, if this is too small, you can click on the YouTube button and then you can watch it on YouTube. Okay? And then that's it. 3 questions. And you press next page. And then there are more videos to watch. Okay, so this is me doing the experiment. You would like my in-person class is doing this next week, right? But obviously you can't do it. So I did it for you. You watch what I'm doing, and then you will answer the questions. Okay, see the timer is going at the top right. Okay, so you have to finish this within the time limit. 3 hours is more than enough. Okay, most people would get this done within an hour or hour and a half. Okay, but I give you extra time just in case you want to read through your answers, just in case it takes you a long time to type things. Okay, and then there's a question 4. What is the independent variable in this? How many questions? 20. Okay, you can see right here, 20 questions. So what is the independent variable in this experiment? Okay, if you know it, you can answer it. Okay, but if you don't, if you forgot what independent variables are, standard variable and whatnot, there is a help video. So watch it. It will help you. Okay, and then moving on, there are more stuff, more help video, and so on and so forth. And then, so it's just all about, watching the video, answering the question is not difficult. The only thing that might be a little bit difficult for some of you is this section where you're going to be analyzing the results, okay? So there are some data here, and you have to create a line graph that shows the above data using Google Sheet which is free if you have a Gmail account, okay, or Excel. Actually, Excel is also free for you. You can get it from My Centennial, I believe. Okay, there's a link. It's like a free copy for all the students, I think. Okay, but if you don't want to download it, you can just use Google Sheet, which is online. Okay, so here is a video that shows you how to make the graph. Okay, if you're not sure how to do a line graph, just follow the steps in the video. Okay, even though it's only 3 hours long, if you follow step by step, you should be okay. Did you guys actually learn how to do graphs with your online teach with your computer teacher? Excel graphs from your computer course? Have you guys learned that yet? Yes? Okay. Because some are saying not yet. I guess it depends. Like, I don't know, maybe you guys have different teachers. Regardless, there is going to be a help video right here. It's only 4 minutes long, and you literally just have to follow the steps. Okay? And then it will show you how to do it. Okay, done. To upload your graph, you click on the camera button, which sometimes might not be visible. You have to click on the dot 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 here. Actually, where is it? My goodness, what if I make it bigger? Oh, I don't know why it is not here. Maybe they changed it. Sorry, I don't know what they did to the camera button. But this is how you can do it. Okay, you can go insert stuff, and then you can upload it from your computer. Oh, actually, no, it's not. Oh, they took away the camera button, which you can just upload directly from your phone. Let me see again. Insert image. Yeah, and then you can get it from your computer. Okay, so you can upload your graph that way. Okay, you can take a screenshot of your graph as well. If you're using PC, there is something called snipping tool. Right? And you can watch this again in the recording, right? There's something called a snipping tool. And it's loading and loading. And once you have the snipping tool, you can click on new. And then you can just highlight whatever part of your screen you want to capture. In your case, you'll be capturing the graph. And once you've done that, you see I just captured my screen, right? And then you can save this on your computer, and then you can upload it. Okay? So that's the only part that tends to take a little bit longer, creating the graph. But like I said, 3 hours is plenty of time. And then let's see if there's anything else here. There are some data analysis. Yeah, you can copy. I think you can copy and paste. You can try. Okay, see if it works. Okay. Don't you guys have to do this for math class, like for your math evaluations? Like you upload your solutions to your quiz or something like that? Or am I mistaken? Yeah. Okay, so like you would know, right? It's the exact same concept here. Okay, and then there are some data analysis and conclusion questions here. Okay, so 20 questions in total, 3 hours. It's a very easy lab. And then when you're all done, you click submit. Okay, and it will tell you whether you're missing any answers or not. I'm just gonna submit it because otherwise I can't access other things. And that's it. Okay, so you can do this between now and next Wednesday. Okay, since we don't have class on Monday, okay, maybe you want to do it on Monday, maybe you want to do it Tuesday. Doesn't matter. Any questions before I let you go? Great. If you have any concerns, of course, email me and I'll be happy to help you. Other than that, I will see you all next Wednesday. Have a good week, guys. What is the way of the lab? Oh, 10%. Okay, 10%. Lab is 10%.
Bye. Thank you.
Okay, thank you. Bye-bye.